Kia ora koutou. Hi everyone, I'm Kerry Hilligan and I lead the Tissue Immunity team at the Hilligan Institute of Medical Research right here in beautiful Wellington, New Zealand. In our lab, we study immune memory as a function of whole tissue and networks of cells, and have a particular interest in understanding how inflammatory or infectious history of a tissue impacts host susceptibility to subsequent infection or disease. This is important because each inflammatory event, infection, or injury leaves a distinct transcriptional, epigenetic, metabolic, and cellular imprint on the tissue that can affect responsiveness to new inflammatory events. Our tissues of interest are the respiratory tract and the lung, where we assess how different microbial exposures or tissue injury events impact the pulmonary environment over time. We then ask how these different inflammatory imprints affect the host susceptibility to infectious disease or allergy. To ask these questions, we employ a systems immunology approach to mouse models of pulmonary inflammation, where we monitor and assess the animal's condition through measurements of body weight, body temperature, and disease scoring. At predetermined endpoints, we collect lung tissue that is divided into three separate processing streams: single-cell suspensions for spectral cytometry and single-cell sequencing, homogenate for cytokine multiplexing, measurement of pathogen loads and PCR, and preserved tissue for histology and immunofluorescence. One of our major interests are tertiary lymphoid structures that form in the lung, otherwise known as inducible bronchial associated lymphoid tissue or IBALT. These structures, shown here in red, develop during resolution of inflammation and facilitate the generation of high affinity antibodies. To study these structures in greater detail, we are using photoconvertible dyes to specifically tag and pull out the cells within the structure for immunophenotyping and sequencing. To hear more about this work and our work around inflammatory conditioning of the lung and host resistance to infection, come along to my presentation on Friday, August 22nd at 1 p.m. See you in Vienna.